Well, first of all, I must say it's a very big conference. I mean, more than 10,000 attendees and more than 250 sessions organized. So it's very difficult, of course, to summarize in a single sentence what's going on in this conference. Almost all aspects and new and emerging issues have been discussed during the last 4 days. If I want to explain in one single word, I would like them to say that I learned something. So that's the most important part. But it's not only learning from listening to the lectures, as I was said. I mean, they meet their colleagues, they interact, they learn from each other, and they will have the feeling that ECCMID is a great venue. So I just updated myself. So I'm now full of idea what's been going on in the world about infectious diseases and clinical microbiology. I think this is the most important thing.
I think they should have the feeling that we contributed to the scientific debate and thought and to an advanced level professional education. That's the one side, and the other side is meeting colleagues, making friendship, having a good time here in Copenhagen, and enjoying their time being here.
My personal view is that the great success of ECCMID is the networking.
The
science is fantastic, the sessions are fantastic, the number of different sessions, fantastic. But I think a lot of people come here because there is so much networking opportunities.
I think it's a one-stop shop. So it has all of the basic science and it has all of the clinical science, and the combination of the two that is required to generate cutting-edge science and care for patients.
It's of course antimicrobial resistance and the solutions to tackle with the antimicrobial resistance. Of course there are sessions about viral infections and fungal infections as well. Now there are bugs in the hospitals infecting patients which cannot be treated by antibiotics.
I have a core belief that we have patients that are currently dying because we aren't getting the exposure of drug right in those patients, so particularly for antibiotics, whereby there's a very strong concentration to effect relationship, that often that concentration that these patients are manifesting isn't as good as it should be. Yep. So because of that, they're suffering and they end up dying. And so all of my research is aimed at trying to arrest that problem.
Absolutely. The cost of these infections is really a big problem because you have to reoperate in many occasions as patients to spend a lot of money because the antimicrobial therapy takes a lot of time, general no less than 3 months. So it's a big issue, and it's important to get together and to find solutions for diagnosis and management.
Unfortunately, I must say that the antibiotics are not the only solution. But as one of the keynote speakers mentioned, that we created these problems with the antibiotics. So antibiotics, at least in part, are responsible for creating these problems.
I was always aware that there was a slow increase in antibiotic resistance, which is inevitable if you use antibiotics. I'm very, very surprised by the recent developments in carbapenem resistance that have emerged so rapidly, and they are coming from such an unexpected area, from the developing countries towards us, which was completely unexpected and unpredicted. And therefore this is a major threat for which we don't have an answer yet.
And then there have been some reports about new antimicrobials. For example, there is a new tetracycline derivative, eravacycline, which works as good as a carbapenem antibiotic in intra-abdominal infections. Well, you might ask, so what is new if it works as good as a carbapenem antibiotic? But the response is, of course, there are now infections like intra-abdominal infections. We have to use carbapenem antibiotics. And if we use carbapenem antibiotics, these are the last resort type of antibiotics. There has been a problem of emerging carbapenem resistance throughout the world, particularly in Europe, in the southern Europe. So if you put them into reserve, if you don't use carbapenems, but instead you use another antibiotic, another alternative, you would say carbapenems, and then that would cause obviously less emergence of resistance.
So we have to look for what we left behind when new antibiotics came in the 70s, which seems to be the miracle. And then we left those antibiotics aside, and now we have to rescue them.
These countries in the Arabian Peninsula are very unique. They have a very unique demography.
Yes.
That in some as much as 80% of the population are expatriates like myself, coming from very different parts of the world, quite often from countries with a very, very high level of rate of resistance on their own. And so that makes these countries in the peninsula very interesting.
What we have done actually in the first day of the conference, that was the UCAS meeting, and this UCAS meeting was broadcasted to China. And in China there were 277 centers, these are centers, not people, have seen these UCAS, because UCAS has become the world's biggest organization about setting breakpoints now. So UCAS has been accepted, UCAS criteria has been accepted everywhere, including United States. But China was planning to be a part of UCAS, so I think this broadcasting was very important for them to get to know about the principles and the work has been done in UCAS. And we have a large, I think more than 250 Chinese colleagues are attending this conference, but China is a huge country, so we were able to reach them. And what we do, of course, we record all these sessions, so we are going to put all these sessions on our society's website, so those of the people who are interested in, who could not attend, but even if you attend, because there are 20 parallel sessions, it's impossible to attend all of them, so you can go to the website and see all these sessions. I think this is one of the most important aspects of the Congress.
This is a quite important point that we clearly understand at the very beginning of UCAS, that we have to share the information. We have to share also how we take the decision of, for instance, the breakpoints, that this is one of our remits, and this is openly available in our UCAS webpage.
This was one of the new things that we experimented at this Congress, actually, the pipeline corner. What we did, and it's just started, we are going to expand it next year in Istanbul. We invited those small-scale companies, these are startup companies in most of the world, but they are doing a huge research on antimicrobials. So I invited them in our pipeline corner.
Because of this Drive AB project, and it's so challenging that we cannot achieve anything without involvement of all stakeholder groups. And one of the main stakeholder groups is small companies that are focusing on antibacterial research and development. So here at ECCMID, we want to give them the opportunity to present their work, and some of them do this. In addition to this, it's really a first this year for ECCMID that we provide a stage for these small companies to present their work. And this work may be the future of new antibiotics.
For the first time, ESCCMID is like opening the doors to the veterinary sector. As I mentioned before, at ECCMID there are only a few veterinary microbiologists so far, but with these initiatives in place, our goal and our vision is to attract many more in the future, and create a forum where veterinary and medical microbiologists can meet and interact between each other on topics of common interest, like antimicrobial resistance, but not only. All the zoonotic infectious diseases will of course be a topic of common interest.
It appears that there are now new ways to modify the emerging bacterial resistance. For example, microbiome modification. This is the modification of the human gut flora, particularly, seems to be very important.
I think there's a lot of people who haven't in the past studied the gut microbiota, who are becoming more and more interested in it now. They want to hear about the latest research. And it's really an area that has exploded very dramatically in recent years.
I think that the concept of that bacteria are always our enemy is wrong. I think that most of the bacteria are beneficial for us, and the whole new area on the microbiome is an example of that. So that concept is changing now. And there are only a few pathogenic bacteria, and will they ever be eliminated? No, I don't think so. I mean, despite all the new antibiotics that we have invented and have tested, no, there's still new mechanisms that can develop in bacteria and make them resistant. And more interesting is, of course, the development of new bacteria strains that contain new virulence characteristics and that can cause huge outbreaks.
Of course, I must emphasize that the diagnostic part of the Congress was very important. Although I'm not a microbiologist, I'm an ID physician, but of course we always collaborate with the microbiologists, and we need them, because if you don't diagnose the disease, what you're going to treat? You're going to use everything empirically, which eventually create resistance. So a large part of this Congress is dedicated to the diagnostics.
We started a project where we want to use genomics in the clinical diagnostic. It's still not yet applied in most diagnostic setting. It's more research-based, but I think in the future it will become a great tool for clinical diagnostic. I think the chlamydia-related bacteria are yet unknown. There is really few people in the world working on that things, and I think there is a lot of things to do. By studying these bacteria that grow much better and faster, it was possible to identify the protein involved in the division of Badlia, and then we extended this work to the protein involved in the division of chlamydia. And all the division protein are putative targets for new antibiotics. And this is also a breakthrough that we published last year.
That's especially important to understand how these bacteria evolve, how they mutate to acquire, for example, resistance, virulence, and also to be able to track their spread. That's some points that we weren't only able to answer these questions by keeping sequencing.
Whereas now with molecular diagnostics, we more and more look at the DNA of the microorganism, so we don't culture anymore. And a good example of that is, for example, the whole microbiome analysis, in which we now are going to look for specific bacteria which we don't know yet, because we don't know how to culture them. So we are now looking on a molecular way on what's in our body. And now we are focusing not only on pathogens, so the bugs which make us ill, but we're also looking at the commensals. It's the problem of the iceberg. We are knowing only the top of it, and that's what we culture. But most of it is below, and we are now diving into the below. And I think that's very new, and I think also will change the way we look at medical microbiology, and we will learn a lot. We have to learn a lot. So it will be a game changer, so to say. It takes some years, undoubtedly, but at the moment I think we are at a perfect moment to change medical microbiology.
This is extremely important, and this is a very strong and important part of the antimicrobial stewardship, because what we are aiming at the moment, the shorter the duration of the diagnosing of a particular microorganism infecting a particular patient is extremely important, because in that case you can target your therapy and you can narrow your spectrum of activity. So early diagnosis has extreme importance.
Nowadays you can detect an infection in hours. But I think that in the future we will speed up this diagnostic, and probably will be that in minutes. The methodologies are changing so fast, and the new methodologies, nanotechnology, microfluidics, are implemented in this rapid test. And as I said, in the future I think that the rapid test will be really rapid.
We are dealing with sudden cardiac death cases, and we try to, as I said, to convince medical examiners and also the judicial authorities of the importance of performing these studies always, because it's a way of learning about death.
We have been discussing whether other ways to fight against these bacteria, not using antibiotics, or in a rational use of antimicrobials. So in that sense, antimicrobial stewardship policies, which includes infection control, which includes vaccines, which includes rational use of antimicrobial agents and shorter duration of antimicrobial therapy, have all been discussed in various topics. For the viral infections, there have been various parts of the conference which is involved in viral infections. For example, there was a session about Ebola, which is still going on, although the epidemic seems to be waning. I must say that we have awarded Médecins Sans Frontières for their heroic efforts they have done and they are still doing in West Africa. And there were speakers from Médecins Sans Frontières and also other people who are fighting these infections in the field. So they gave an excellent conference on the first day of ECCMID. That was interesting because I think this is a clear example what an emerging infection, which hadn't been thought previously could cause such a huge problem in the Western world, could create an emergency in the world scale. So this is extremely important. There have been a breakthrough late breaker abstract session. There was a new vaccine about zoster, which works much better in the elderly because the current vaccines has less efficacy in the elderly patients, and this vaccine seems to work better in elderly patients with two doses, so this is something new.
Despite the advances in treatment, which clearly have been extraordinarily effective to bring down all the diseases that is a consequence of being infected with HIV, we unfortunately haven't been able to stop transmission. And the major challenge that we have globally at the moment is that the number of new infections actually exceed the number of people that we're able to put on treatment. So we are in a certain way behind the ball in terms of handling this pandemic. So we really and truly need new innovative ways of limit transmission so we can start to get ahead of the curve and eliminate HIV from the globe. So it's really a dilemma at the moment of whether we should actually be more aggressive in our treatment approach. And there's a lot of important research going on at the moment to try to elucidate this, because it could well be, we don't know at the moment, but it could well be that actually treatment is also providing benefit to the individual also in the early stages.
So I presented the new vaccine which targets 9 different genotypes, and we believe that with this vaccine we will completely change the way how we screen and how we prevent the cervical cancer. We're now going to the eradication of the cervical cancer, which is something which is unbelievable achievement.
We can do a lot more by actually studying the real life effect of our interventions, and we can save a lot more children in low-income countries, and I say a lot more. With thousands and thousands, probably millions of children could be saved if we vaccinated correctly. If we took the non-specific effect into consideration, we could save a lot of money in high-income countries. If we actually understood, our policies are not based on truly controlled studies. We show this is the best age to vaccinate. We have shown in Denmark that once the children get their measles vaccine in Denmark, then the risk of getting hospitalized with severe lower respiratory infection is 20% lower. That costs a lot of money in Denmark. So you could actually also, you could save lives in low-income countries, and you can save costs in high-income countries.
Then the public understands, but I don't want them to understand in this way, because that means they have to be sick and they have to die in order to understand how important it is. We start to see things that we could dream about, but now it comes to reality. Less hospital burden, less hospitalization in general, less visits of children to clinics. So it's really showing that the new vaccines can change a lot.
We have a problem, particularly in certain countries like in Netherlands. Those antifungals have been used for agricultural purposes. And these antifungals are very similar to what we are using in the human beings as well. So once you use widespread these antifungals in the environment for agricultural purposes, you are creating some resistant fungi, which then in turn can infect your patients or can colonize the patients. And then once they become immunosuppressed, their immune system is destroyed by the chemotherapy or the disease, these fungi can infect these patients.
Because if you look at the high-income countries, then of course we do need better diagnostics. We know that probably around a quarter of our infections are not timely diagnosed, and that we see raising incidences of many of the invasive infections that have high mortalities. And particularly that we really see raising incidence of resistance. And we see that in Aspergillus fumigatus, where it's azole resistance, and that is not mainly due to the human use of azole medication, as most other resistance, but it's due to the massive use of aerosol pesticides in agriculture and for material preservation.
The diagnostic procedures are still poor. There are very interesting study reports out there looking at autopsy data in hem-onc patients or hematological malignancies, that those patients have 30% have a fungal disease, but only 25% of those diseases are recognized prior to death.
Wow.
Meaning we're missing a lot of these patients.
And Istanbul, I think, is a fantastic place. ECCMID has now become the biggest conference of the world about clinical microbiology and infectious diseases. And the science, the quality of science that we are showing, quality of the scientific program is good. And we plan, of course, a top quality of scientific program in Istanbul as well, which will attract many scientists throughout the world.
Medeniyetlerin birleştiği İstanbul'a hepinizi ECCMID 2016'ya bekliyoruz. ECCMID 2016 İstanbul'da.
Everybody's invited for Istanbul.